And I'm super excited to be here and talking about my work in this seminar series because I got introduced to it not that long ago and it's been outstanding so far. So I'm really excited to be here. And there we go. So I thought I would kind of get started here with just a big picture of the themes that underlie my work so that gives you some context for what I'll be talking about. And because I wanted this to be kind of a big picture, I made it a big picture. So I actually work on molecular modeling, and I'm very interested in how molecular movement and interactions between macromolecules are influenced by their shape and how this actually dictates how they function. And I focus particularly as a modeling person on two aspects of the systems I study. And the first one, and the one that we're going to be focusing on in this talk, is the geometry and topology that's associated with these molecules, as well as stochastic modeling of the interaction and dynamics that govern how they change conformation. And so those are sort of the big themes underlying this work. Oops. There we go. So the problem I'm going to be talking to you about involves what's called DNA replication stress. So DNA replication is exactly what it sounds like. It's where the double-stranded DNA in a cell is copied quite faithfully to produce daughter strands that then can go on into making new cells and so on. But there's ways that this process can go wrong. And one of them is through these very odd non-B form secondary structures. And these can disrupt the molecular machinery. It's called the replisome, all of the parts and pieces that actually carry out replication. And so to do this, we're actually going to be looking at this from a geometric perspective um, on how the structure of those G-quadruplexes, which I'll be calling G4s in this talk, how they mess things up in DNA replication. So in order to talk about how DNA replication goes wrong, it helps to kind of understand a little bit of how it goes right. And what happens is DNA replication stress is really about fork stalling. And what that means is there's these three main stages of DNA replication. There's what's called licensing, where the origins start gathering the machinery. There's when the origins fire, which is assisted by a particular replication complex, an initiation complex, and then there's fork progression, where the two main hexamers that I don't know if you can see my mouse here, but we have these two hexamers will start separating, and it forms these two forks, and they're moving away from each other bidirectionally. But through this whole process, you have different proteins that are coming in and out. To affect this very complicated process and affect it in a very exact way. And I thought the best way to kind of illustrate this would be to just show you it in action. This is a beautiful video from Cold Spring Harbor Labs that kind of illustrates, and I'm going to kind of talk through this for you for how this actually proceeds. And so we're going to turn the sound off there. But basically, we have double-stranded DNA. The blue thing here that's really cranking it out is the helicase complex you're going to be hearing a lot about in this talk. As you might know, DNA has, when it's unwound, it has a leading strand and a lagging strand. So the leading strand pumps right into a polymerase. The lagging strand has to go through this complicated spinning off these loops that are then replicated to create double-stranded DNA. But the real thing orchestrating it at the very front is this helicase complex. And you're going to be seeing a lot about that in this talk. I'm glad that video worked because it's so lovely. So the thing that I'm going to be talking about is one of the primary drivers for replication stress in DNA. Is what's called G-quadruplexes. Okay, these are sort of funky forms of DNA that happen quite naturally and transiently, and they're going to be the main stressor for that replication process that happens. And what these are is these are these four-stranded non-B form DNA secondary structural elements that form in the single-stranded DNA as it's coming out of the or going through the helicase, and it forms from G-rich regions because it's the G um, motifs that form together to form these tetrads through Hoogsteen bonding, which is sort of a non-standard hydrogen bonding form, and it forms these little plates, and then those plates stack up that in a way that um, allows them to come together and form these secondary structures, these fairly bulky secondary structures out of these stacked plates. And these are typically stabilized using a monovalent cation of some sort, and typically potassium. So, um, and what we're interested in this talk is the topology of those strands. And so this is a schematic I put together of some of the main types, and there's kind of a zoo of the different ways that these um, G quartets can come together transiently, either from unwound single stranded DNA or even from double stranded DNA that has transiently melted to form single stranded DNA. But what happens is that you have different ways that they can line up. And the ones that we're most concerned with are these parallel, anti parallel, and then a hybrid form. So the regions between these are the G tracks, and then I'll have loops that kind of connect these Gs. But these are all guanines that are on the DNA strand that are coming together to form these very particular structures. And so, go to the next slide. So what this means is there's lots of different shapes that these particular secondary structures, these non-standard DNA forms, can take. And so I'm showing you just three kind of representative ones from the PDB. So these are solved structures of these um, these different DNA topologies, and they're in the parallel, anti-parallel, parallel, and hybrid forms here. And just by looking at them, and you can see all those G's lining up and forming those stacking plates, but which are characteristic of the G quadruplex. But what you can also see when we look closely at this is when we think about this from a space filling view, then it's very clear that they're occupying very, very different shapes. And one would anticipate that this would influence how they interact with the replisome and the replication machinery to cause havoc there. And so what happens is we have studied these G quadruplexes for some time, and they've been heavily studied just with sequencing methods to kind of look in the genome for what are potential regions where we find these G-rich zones that could form these transient secondary structures. And so there's a lot of work done looking for just their distribution throughout their genome, but their exact role and the mechanism of how they kind of cause disruption and whether they have positive roles as well is still very much a question um, in the field, and that we're really kind of trying to work out, what these are, because many of these are very disease associated, particularly in cancers. And so we want to be able, and that makes them potentially targetable for mechanistic interference. And so we're going to be looking at how these things actually play out inside of the replosome. And so our main question, just to make it quite clear what I'm talking about, is I'm very interested in how the structure, as in the shapes of those G4s, can dictate how it disrupts DNA replication, and how we're going to use to do that information about structures where these things are present because that allows us to look at them geometrically, and get some information about what that interior space of the various proteins in the replosome, what those macromolecules look like kind of on the inside, and how these things might be affecting the types of disruption that they do. So I'm going to step aside for just a moment to talk about the types of geometric modeling that are at the basis of the work that you're going to see. And so I don't know how much people are familiar with computational geometry and Voronoi diagrams, so I'm going to take just a brief digression here to explain a little bit about what's going on. So if I have a collection of points in some in RD and D-dimensional space, okay, we're going to be thinking, in the case of the protein modeling, we're going to be thinking obviously about R3, the three-dimensional space. All the pictures I'm going to show you, just to kind of keep things clear, are going to be in R2. But what we want to think about is we have this collection of points. Okay, we're going to call that S. And what a Voronoi tessellation does for us is it partitions the space um, around that set of points such that each cell contains only one point and is bounded by a polygonal region of locations that are closest to that point in the rest of the space. And so if that was a little bit too much of a mouthful, I just kind of have a nice formula here that just illustrates that what this means is that if for my point S, if I pick a point like right here and call that S, then all of the points in its Voronoi cell are closer to it than to any other point in any of the other um, um, points in the space. And so what I can do is then box off the region of those points as a cell that belongs to that point S. Okay. And so that's really what a Voronoi diagram um, gives us. It partitions the space into these polygonal regions. Okay. Um, so there's a dual thing to this called a Delaunay triangulation, or in the case of 3D, that would be um, a tetrahedralization. Okay. And what I'm going to do is I'm going to take my Voronoi diagram, which you can kind of see in the lighter color in the background, and any point that 2 points that share a Voronoi edge, okay, I'm going to draw a line between them. And what that's going to do is partition the convex hull here into a space of triangles, okay, or tetrahedra in 3 dimensions. So that's kind of the main gist of what a Voronoi diagram is in terms of representing how space is partitioned by a set of points, okay? And this works just analogously in 3D or in any D for that matter. So that dual Delaunay tetrahedralization in 3D connects points in S where the um, um, the two Voronoi points share a plane, as in there's a line dividing them, and along that line dividing them, I'm equidistant between the 2 points that are contributing there. Okay, and but I can take the same set of points. By the way, this isn't exactly the same set of points as the one on the previous slide, but I can take a set of points and just form a Delaunay triangulation on it by taking any 3 points and drawing a circle around it and then finding the center of that circle. I can take these 3 points, I can draw a circle around it and find the center of that circle. I could take these 3 points, blah, 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 draw a circle around it, find the center of that circle. What's interesting is all those centers, when I hook them up, give me my Voronoi tessellation. Okay. So these are just two versions, and I can essentially computationally interconvert from one to the other. If I start with a Delaunay triangulation, I can find its dual Voronoi tessellation, if I start with the Voronoi, I can find the dual Delaunay. And so the question becomes, why do we want to do this? Okay. And the reason is these are very, very computationally tractable objects, even for very large systems. And you can imagine, just from what you've seen of some of the bigger proteins that we were looking at earlier in the talk, that we're talking about a lot of atoms in these systems. But there's very rapid, very stable algorithms that enable us to do very accurate computation in these sets. And so they give us very efficient representation of 2D and 3D point clouds. And in our case, our point clouds are going to be clouds of atom locations in 3D space that we have from structural data. Okay. Once we have those representations, they're very nice to kind of manipulate to be able to use for the types of things that we want to do. And I'm focusing on one particular algorithm called the mole algorithm. And what this is going to do is it operates specifically on molecular structural data, and it finds pathways along those Voronoi edges that minimize some sort of a cost function, and it allows me to locate pores and pockets inside these structures. And so I can compute molecular surfaces or interior cavities by pruning down the various tetrahedra that are formed from this tetrahedralization, which is the dual of the Voronoi diagram. Um, and I'm going to use particular sphere radii that are related, by the way, to kind of van der Waals radii, and work with the connections between those tetrahedra to try to understand the shape of that space and to locate regions of interest like channels and tunnels that might be in the molecule. And that's how we're going to be using it. And so just kind of a one last kind of idea about how this is done is that if I have my sort of cloud of points and my Delaunay, excuse me, in this case, my Voronoi um, characterization of it, I can put weights on the different faces and edges of my um, Voronoi diagram that effectively um, downweight things that are farther apart. And so these two things share this face, and I'm going to give it a small weight because they're very far apart. These two things share this face, and it's going to get a large weight because they're quite close to each other in the Voronoi diagram. And so this allows me to try to locate these paths of small value weights as potentially zones that are voids in the space. And so that is the main motivating idea behind how this geometry allows us to find these things. So to take this to a concrete kind of example now, there was recent work, it's absolutely beautiful work. I highly recommend this paper by led by Batra in the Remus group that reported last year on some cryo-EM structures of just the helicase complex part of the replisome. Okay, and they were able to find it in actually interacting with these G quadruplexes. And remember, those G quadruplexes come in different geometries and different topologies and flavors. Okay. So I'm focusing on two structures here. These are done with cryo-EM, and this was absolutely groundbreaking work. And they did a tremendous amount beyond the structures, biophysical measurements, and so on and so forth. Um, and what they were able to show is they were able to kind of catch in the act one of these G quadruplexes literally getting stuck inside of the channel of this replicon or of this replicon element, in particular of this hexamer that is part of the helicase. Okay. And they've got structures here. They're at about 3.2 angstroms and 3.5 angstroms. they actually caught two different orientations of the G4 in one of the pockets here, which is just remarkable when you think about it. Um, and what's important to realize, though, is once the G quadruplex is parked in there, it is no longer accessible to the types of tools that the cell has to try to unwind those things um, and make it so that the process of machinery can keep going. Okay. And so we have these PDB structures of where these things are. And the way I'm actually doing the modeling is I'm actually taking these structures. You can see the two G4s in here. They've actually removed M4 and M7 from the top of these. These are channels that are surrounding the DNA. Um, and I strip out the DNA so that I have just the channel to work with, and I'm using these computational geometry tools to characterize that. So they were able to show that basically a single one of these G4s is sufficient to stall the replosome, particularly in the yeast cells, although they did work with human cells. They show that it actually takes different orientations in that channel. Um, they show that it stops that CMG, which is the three kind of main components in the structure, from doing that translocation. And one of the things they conclude from their biophysical work is that it's really not dependent on G4 topology. But one of the things that I was emphasizing earlier is that those topologies are, in fact, quite different. And so this is an ideal system to study with the computational geometry. I'm going to very quickly roll through this in the interest of time. These are three different views of the original structures. You can see here the DNA kind of going through this main pore. I've kind of twisted it here so you can see that it's coming in as double-stranded DNA. It will become single-stranded within the helicase. And in this view, you can really look down that DNA and see those stacked G structures forming the G quadruplex. And so we actually carried out pore analysis using the Voronoi and were able to locate the pores that, in fact, this is passing through. I'm going to show you two views here. Remember, I said there's two states of the G quadruplex in here. State one is on the left, state two is on the right, and we're kind of looking down from where they're entering. Um, the complex. But if I rotate this about 90 degrees, I can see the length of that channel. And I can actually measure using kind of the information I got from the Voronoi representations. I can measure different parts of the channel. I can measure channel width, properties inside the channel, by the way, and so on and so forth. And what was beautiful to see is this really captures where the DNA is going, the double-stranded coming in here, right around in this region, and this one here is where that thing is getting lodged. And then the rest of the single-stranded DNA ahead of that G4 is exiting through this part of the channel. And so this is a wealth of information about what's actually going on with how these G4s are getting caught, because we can see quite clearly that the dimensions of these channels. And so this is just briefly a channel profile. The first channel was about 142 angstroms long. Second one, this is, by the way, the end where the double-stranded DNA kind of butts up against the helicase and it's unwinding it into single-stranded DNA. And we see definite regions where there's constrictions. So this is the width of these radii, the radiuses, and they're broadly similar, but we definitely see different behaviors in terms of the size, particularly of those pockets where the G4 would be lodged. And so I just want to close here with a summary of this. So I think that these geometry and Delaunay tessellation tools are actually a very powerful representation that allow us to ask quantitative level questions about this. Using the beautiful structural data, we were able to locate and characterize that key tunnel that they were able to see. in the CMG helicase complex, the one that gets blocked by the G quadruplex, and that there are really observable differences found, particularly in the region that is occupied by that invading G4. Our ongoing work with this that I didn't quite get finished for this talk is that we're very interested in then taking these uh, geometric structures and docking into it the different forms of the G quadruplex. These were all done with parallel, looking at the anti-parallel and the hybrid to kind of see if those do cause changes in what we would expect for the formation and stoppage of those things. And there's so many more questions I'm working on with this. This is a very rich data set and a very rich modeling setting. So I'll be kind of continuing this for some time. So I do want to acknowledge groups that I work with. The team in the lab that I work with at UB in the National Crystallization Center in particular. Katie Spoth is our cryo-EM person, and she keeps me honest about all the cryo-EM results. And I have been long time collaborating with Joyce Ohm at Roswell Park Cancer Center on topics related to DNA replication stress. So her and her whole lab have been very inspirational for these things, and I want to thank my funding sources as well. And I'm happy to take any questions.